I'll use the mouse. Okay, better put it here. Okay, well, whilst the talk is loading, I'll just say thank you to Dan, to Inbal, and to Karen for the invitation. It's great to be back. No.
Not this one?
No. It's on the hard drive. It's in a folder called Day 2. There you go. That's it. Okay, yeah. It's great to be back. I am sorry that I wasn't here earlier. I think we had a signal transduction malfunction in Newark Airport. We had a gate change just before we boarded the plane, and this information was not relayed to the baggage people who loaded the flight to Delhi's baggage onto the flight to Frankfurt's baggage. And so we had to turn back, deplane, and they switched the baggage. And that unfortunately led me to miss my connection, but I'm glad to be here. And I'll be here for the week, so I look forward to meeting everybody. So as Karen said, my lab is largely interested in roles for microRNAs in development, in aging, and in diseases of aging. And we focus mainly on cancer just because of its accessibility. And I thank Ronit for giving such a great introduction to all the things that I'm going to tell you about today. What I hope to do is convince you that microRNAs play a causal role in cancer, and we've done that in a few ways. First of all, showing that microRNAs control certain key cancer genes. Secondly, by showing that turning on microRNAs in mouse models leads to cancer, and that those cancers can in fact become addicted to the microRNA genes. But I make two disclaimers. First of all, I'm not an oncologist. I'm a developmental biologist, and so we use many of the tools of developmental biology to try and understand these processes. And second, I'm not a chemist or an engineer. So I've had wonderful collaborations with bioengineers who've helped us design some of the particles and some of the nano delivery techniques that I'll tell you about today. And in particular, I'll shout out to Mark Saltzman here in the Department of Biomedical Engineering at Yale University, and Andy Bader and David Brown at a company called MicroRNA Therapeutics, who've provided us with some of the reagents that I'll tell you about today. Let me just make sure I can get this mouse to work. There we go. Okay, so you've already heard, I believe yesterday and already this morning, that microRNAs are intimately associated with cancer. Some of the reasons why we believe that are shown on this slide. I'm not going to spend much time on all of these, but just point out briefly some of the reasons why we think that microRNAs play such an important role in cancer. First of all, we know that microRNAs can be located in regions of the human genome that are deleted or amplified in cancer. Some work done in the early 2000s by Carlo Croce's lab showed that almost half of all of the human microRNAs known at that time, which was about 150 or so, mapped to regions of the human genome that were either breakpoints in cancer, loss of heterozygosity in cancer, amplifications in cancer, and in fact could explain, in some cases, decades-long mysteries as to what was the cancer-causing gene in some of those regions. So our lab and other labs showed at about the same time that microRNAs were frequently misexpressed in cancer. So if you compare normal adjacent tissue to tumor tissue, and this is just one example of an early microarray in this area, you can see that a number of microRNAs are upregulated in cancer and other microRNAs are downregulated in cancer. And I will give you one story illustrating both of those cases today. So the microRNAs that are upregulated in cancer, we sometimes think of as oncogenes or oncogenic microRNAs, and our hope is that we can destroy those microRNAs or inhibit them in some way and lead to a relief of the cancer or a cancer therapeutic. In other cases where a microRNA might be downregulated in cancer, we often think of those as tumor suppressor microRNAs. And the idea here is that we can restore or replace those microRNAs in the cancer and have the same effect. i.e. A potential therapeutic in cancer. So more recently, it's now known that some of the genes that are involved in producing the proteins that process microRNAs can be mutated in cancer. And lastly, and this is, I think, the part where my lab has really played a more significant role than in these other areas, is that microRNAs regulate oncogenes, and they regulate tumor suppressor genes. And in fact, they can act themselves as oncogenes and tumor suppressor genes. All right, so just a brief history of microRNAs from bench to bedside, if you will, or from discovery to bedside. It was only in 1993 that Victor Ambros identified the very first microRNA, Lin4, and for this, he and Gary Ruvkin got the Wolf Prize just, I think, 10 days ago or so. And then when I was in Gary's lab, I was part of the group that discovered the first human microRNA, which at that time was Let7. That was discovered only in the year 2000. So it's been a very short amount of time from the discovery of the first human microRNA to the first clinical trials with microRNAs, which started just last year. So in 2002, the Croce lab showed that, in fact, certain microRNAs were deleted or lost in cancers. In 2007, it was discovered that there were microRNAs downstream of important tumor pathway genes like P53. And in 2011, the first antimer entered clinical trials. Now, it wasn't for cancer in this particular case, but for hepatitis. Work by a company called Centaurus showed that an antisense to a microRNA involved in hepatitis replication is a useful anti-hepatitis drug. And then in 2013, a company called MicroRNA Therapeutics initiated the very first human clinical trials with microRNA replacement. And this is with a microRNA that you've already heard about called miR34. All right, so this was a brief report about the start of that trial in Nature that happened last year. And so just about 6 weeks ago at the AACR meeting, the results of that phase 1 trial were reported, and the phase 1 trial showed limited, if any, toxicity associated with a nanoparticle delivered miR34 mimetic in human patients. So phase 2 trials will start soon. All right, so as I mentioned earlier, I'm going to give you 2 different stories today. In the first, I'll tell you about microRNAs as therapeutic targets. So as targeted therapeutics, actually using the microRNA itself as the therapeutic. And then in the second story, I'll tell you about microRNAs as therapeutic targets, so actually targeting the microRNA as the therapy. And we've discussed some of these approaches and their route to the clinic in a review that we wrote about 2 years ago. So I won't have time to discuss all of the different cases today, but if you are interested, you can go back and get this review, and it'll tell you more about some of the other approaches that our lab and other labs are taking. And I won't have time to tell you about targeting microRNA complementary site variants as an additional approach for cancer. But some of you will know that there are mutations within microRNA binding sites in certain cancer genes like KRAS, like PTEN, like BRCA1, that seem to lead to misexpression of those important cancer genes because they break microRNA regulation of those genes, and we can target those particular sites with siRNAs. All right, so the first story I'll tell you about is microRNAs as targeted therapeutics. And this is a case for delivering microRNAs as therapeutics in cancer. So this is work that I've done in collaboration with Andy Bader and David Brown's lab at MicroRNA Therapeutics. Sorry, when I said I should say our lab. And this is mainly work of a number of talented postdocs, including Feng Trang and Andrea Kaczynski, who thought that potentially we could use microRNAs as a way to knock down oncogene expression in a mouse model or even in human cancers. So this work was initiated by Aurora Kershaw when she was a postdoc in my lab. Okay, so I'm going back to that original paper from Carlo Croce's lab back in 2004, where they showed that a number of different human microRNA genes mapped to regions of the genome that were lost or amplified or otherwise disrupted in cancer patients. And I should say that this paper came out in 2004, but if you look at this list of microRNAs here, it's incredibly prescient. This work predicted almost with 100% accuracy that these particular microRNAs were going to be important in cancer. So some of the microRNAs that I'll tell you about today include LET7 and MIR34, which are two microRNAs that are found on this list. So, for example, LET7G is found in a region of the human chromosome that's deleted in lung and breast cancers, and there are other LET7 family members that are found in this, also on this list, to be deleted in those types of cancers. And this is an example of MIR34A, which is found in a region that's known to be deleted in lung and breast cancers as well. So for the purposes of this talk, I'm going to focus mainly on lung cancer for this part, but we're also working on the breast cancer portion of this. And our hope is that since these two microRNAs are deleted in these cancers, well, first we wanted to show that they were in fact causal or that they played a role in these cancers, and then try to restore the expression of these microRNAs in these cancers. All right, so I'll remind you that non-small cell lung cancer is a genetic disease, as is most types of cancer. And there are a number of different recurring mutations in a subset of oncogenes and tumor suppressor genes that are recurrent in non-small cell lung cancer patients. So near the top of this list are mutations in the oncogenes RAS and MYC and EGFR. You've heard about some of those already at this conference or at this workshop. And then, of course, deletions or loss of tumor suppressor genes like p53 and retinoblastoma. Now, I won't have time to go into all of the background on this, but my lab and other labs have shown that microRNAs regulate many of the genes on this list. And in particular, we showed earlier that LET7 microRNA regulates multiple members of the RAS family, including KRAS, which is one of the most important or one of the most mutated oncogenes in lung cancer, as well as the MYC oncogene, which gets amplified in this cancer and a number of other cancers. And then through an indirect route, we also showed that LET7 can activate the p16 tumor suppressor gene. And then our lab and other labs also showed that MIR34, one of those other microRNAs deleted in this disease, regulates some of the other genes on this list, including Bcell2 and MET, which is another oncogene that should be in this part of the table. In addition, p53 directly upregulates the expression of MIR34. And in fact, some people have argued that MIR34 is probably one of the most important downstream genes of p53, because it regulates some of the factors that p53 is known to affect when it gets lost in cancer. All right, so our previous work had shown that if you systemically deliver LET7 or MIR34 in a lipid nanoparticle into a mouse model that has a mutation in KRAS, you could suppress the hyperplasia that you see in this KRAS mouse model. I won't have time to show you that data, but that was published back in probably 2008, 2009. And we also showed that if you deliver an antisense molecule to these microRNAs intranasally right into the lung of these mice, you can exacerbate their cancers. So showing that these were, in fact, tumor suppressor microRNAs. So some of you will know that in many cases, non-small cell lung cancer patients present with not only activating mutations in oncogenes, but also loss of function mutations or dominant negative mutations in tumor suppressor genes. And in fact, a particularly lethal combination is the KRAS p53 double mutant non-small cell lung cancer patient, which is essentially resistant to almost all therapy and has a very short survival time. In fact, most non-small cell lung cancer patients, only about 15% of them will survive more than 5 years. So this is a very lethal disease, and obviously needs better therapies and a better understanding of the disease in order to combat it. So in order to ask whether our combination or set of microRNAs here, LET7 and miR34, could be effective against this very lethal combination of KRAS and P53 mutations, we built or got from other groups transgenic mice that had mutations in either KRAS or P53 and combined them into a double mutant. And we used 2 different alleles of P53. The first is just a fluxed allele which deletes the P53 gene. And in the second case, we used the mouse version of a dominant negative version of P53 that's commonly found in human lung cancer patients. This is the R172H heterozygous mouse. So you can see that, that these mice have a much shorter survival than do mice that are just mutant for KRAS. So in order to activate these alleles in the lung of the mice, we can deliver an adenovirus expressing Cre recombinase directly into the lungs of the mice, either through intratracheal or intranasal delivery. And when that happens, we see that the double mutant has a very short lifespan. So we asked a few questions. First of all, could delivery of the microRNAs at the same time as we're activating these cancer genes and knocking out the tumor suppressor genes have any effect on initiation of tumors in this mouse? And so to do this, we essentially delivered the microRNAs at the same time as we knocked out the genes, or very shortly after knocking out those genes. So before I go there, let me just show you what happens in this KRAS P53 mouse, because it is, while not perfect, a pretty good model for non-small cell lung cancer progression in humans. Why I say it's not perfect is that it may be a little bit too aggressive for even the human disease. Okay, so if we infect these animals with AdCre at this time and then essentially harvest lungs at different times during the progression of the disease, you can see that by 10 weeks we already have evidence for adenocarcinoma forming in these lungs. By 13 weeks, 16 weeks, and 19 weeks, you can see that there are multiple tumor nodules that have formed in these lungs. And this is really where this mouse model shows itself to be almost too aggressive, because most human patients present with maybe 1 nodule. But here we may have 20 nodules within 1 lobe of the lung. You can see that in this case, there's 1 nodule that's grown to be massive size. And so over time, you can see that there's a time-dependent increase in tumor burden in these animals, and we measure tumor burden as basically the percent area of tumor relative to the area of the entire section. And then we average that over a number of sections, averaged then over a number of mice. And you can also see that there's a time-dependent increase in the number of adenocarcinomas that can be detected in these mice. Okay, so these mice also show low levels of expression of some of those microRNAs that I mentioned earlier, like miR34 and LET7. Not too surprising, miR34 levels are low in this animal, and I told you earlier that P53 directly activates the transcription of these microRNAs. I'm not going to show you LET7 now, but we also know that LET7 levels are low in this mouse as well. Interestingly, some of the oncogenic microRNAs like miR155, which I'll tell you about in the second section of the talk, are elevated in this mouse model. And this is a potential oncomer or oncogenic microRNA. In addition, we can see that in this mouse, some of the miR34 and LET7 target genes like MET and Bcell2 are in fact elevated. So when miR34 levels are low, these oncogenes get overexpressed. And that's also not too surprising based on what we know about the biology of these microRNAs. All right, so what happens if you deliver the microRNA at the same time as you're activating the KRAS and knocking out P53? So we essentially infected these animals with AdCrE, and then the next day we came in with a lentivirus expressing, in this case, just miR34, much like what you heard about from Ronit earlier this morning. And then at 19 weeks, we sacrificed these animals. And as I showed you earlier, at 19 weeks, there is massive tumor burden in these mouse lungs. And we saw something that was quite remarkable. So in the control-treated mice, they have multiple tumor nodules, as I told you earlier. But in the mice that were treated with miR34 at the same time as P53 was knocked out and as KRAS was activated, we saw no evidence for tumors forming in these mice. So miR34, when delivered at the same time as P53 was deleted, completely cured, if you will, or prevented tumors from forming in these particular mice. Now in this case, as Ronit said, this is a very artificial case. You cannot think yet, at least in the Western world, of treating patients with a lentivirus expressing a microRNA. Although I believe that there are clinical trials in China with lentiviruses expressing P53. But we've tried to deliver these microRNAs in a way that could be taken systemically and in a non-viral delivery mechanism. And I'll tell you about those methods in a few slides. We also asked whether waiting for these animals to actually first present with adenocarcinoma and then treating them with the miR34 lentivirus could reduce the tumor burden in these mice. And in fact, you see that that's the case. While the control animals had extreme tumor burden, the miR34 treated animals had very low levels of tumors. All right. So since cancer is a multi-genic disease, and since we also heard about earlier that there is massive tumor heterogeneity, we wondered whether combinations of microRNAs, and in particular tumor suppressor microRNAs, might be more effective than just delivering single microRNAs. So to remind you, we'd shown in previous work that delivering either LET7 or miR34 to the KRAS mutant mouse was sufficient to cause tumor regression, or at least regression of the hyperplasia that occurs in those animals. But now we wanted to know whether delivery of these microRNAs systemically could show any efficacy against the KRAS P53 double mutant mouse, which is extremely aggressive and fails all different cancer therapies that have been tried. In fact, I'm told that Tyler Jacks has I think a $10,000 reward for anybody who can cure this mouse. So the challenge is on for you guys to get out there and cure this particular mouse, because if you do, A, you get $10,000, B, you will help a lot of people. Okay, so just to remind you that that non-small cell lung cancer has mutations in multiple different cancer pathways, and that LET7 and miR34 affect multiple of these different pathways. And so we asked whether the combination might be better than each microRNA on its own. So the only things that have changed in this particular section of the talk relative to what I told you earlier is that we've used a combination of microRNAs. And when I talk about a combination here, it's half the dose of each microRNA, so that the total dose of microRNAs given to these animals is equal to what we would have given with just miR34 or LET7 alone. We've used a more aggressive model of non-small cell lung cancer. In this case, we've used the fluxed P53 allele. And the microRNAs are, in fact, delivered systemically. So they're delivered through tail vein injection using a nanoparticle to encapsulate and protect the microRNA mimetics. Okay, so before I show you what happens in the mouse, let me just tell you what happens in vitro, in cell culture. So we identified from the NCI 60 panel of non-small cell lung cancer lines lung cancer lines that were mutant for both P53 and for KRAS. An example is shown here, like this H358 cell, which has a G12V mutation in KRAS and a deletion of P53. And so basically what we found was that in almost every case that we looked, the combination of LET7 plus MIR34 worked better to prevent or to slow the growth of these double mutant lung cancer lines than did either of the microRNAs alone. So once again, half the dose of LET7 plus half the dose of MIR34 was better than the full dose of MIR34 or LET7 in most of these cases. And we calculated a combination index. For some of these cell lines, it showed that this combination of the two microRNAs was in fact synergistic. Our most dramatic effect was seen when we looked at migration of these cells just through a transwell migration assay. And you can see that the combination of the two microRNAs almost completely prevented cell migration in this metastasis model. In addition, we noticed that the combination of half the dose of each of these microRNAs was better at knocking down more oncogenes than just single microRNAs by themselves. So just a few examples here. If you look at adding MIR34 to A549 cells or H441 cells or H23 cells, we can see that MET gets downregulated in all of these cases, and that's a known target of MIR34. In addition, if you add LET7 to these cells, you can see that a known target of LET7, which is Lyn28, another oncogene which has just recently been identified, is knocked down by LET7. And in fact, MYC is also knocked down by both LET7 and MIR34, which is also what we had shown before. Oh, sorry. Ooh. How do I go back? Nope. However, if you add both LET7 and MIR34, you can knock out MET, Lyn28, and MYC all at the same time. So we also characterized this lung cancer model for expression of microRNAs. Much like what we'd seen in the other allele of P53 in combination with KRAS, these mice also have very low levels of MIR34 and lower levels of the LET7 microRNA compared to uninduced littermates from the same litter. And in addition, they overexpress certain oncogenic microRNAs like MIR21 and MIR155. So how did we deliver these particular microRNA mimetics? So much like what you heard about in the earlier talk this morning, we have partnered with a group that has developed a lipid nanoparticle that targets to the lung. It also targets to the liver, but in our case it was very important that we used a nanoparticle that was able to also target to the lung. So this particular nanoparticle is nicknamed a smarticle. I have to say that I think that's not a very good name. It's not that smart. I think it can be way smarter, as you heard about in the earlier talk from Ronit. We're just beginning to make these things smart. However, it does have some very attractive features that made us want to use this particle. First of all, it's already in clinical trials for delivering siRNAs. It's been FDA approved for use in humans, and so that made it very attractive. We wouldn't have to go and get approval for any new kind of nanoparticle that we wanted to use in a clinical trial. So it's a neutral lipid nanoparticle at tumor pHs. So once again, the hope is that it delivers more effectively to tumors than it does to normal tissue. Sorry, I think I've already mentioned this already, but it has now been used in phase 1 trials to deliver a microRNA mimetic MIR34 with very low toxicity. So the idea is to use a microRNA mimetic that's loaded into this smarticle, nanoparticle, and deliver that to mice and to patients. So we did the preclinical work in the mice. This work is in revision, so it should come out quite soon. But the basic idea here is that we activated KRAS and deleted p53 in that very aggressive model of lung cancer. We had to, in this case, we could only wait 7 weeks in this case because this model is so aggressive that adenocarcinomas have already formed within 7 weeks. And these animals are almost dead by 12 weeks. So we delivered into the tail vein of these mice at 7 weeks either a control microRNA mimetic, which is just basically a scrambled sequence. We delivered Let7 by itself, miR34 by itself, or half the combination of each. And that's what I've called dual over here. We did 14 injections, so every 2 days for 28 days, and then we stopped the delivery. And then waited a few days and sacrificed the animals. So just to show you that, that this particular smarticle nanoparticle can deliver microRNAs to the lung, we can detect increased levels of miR34 in lungs from animals that have been treated with this particular nanoparticle. We also see increased levels of miR34 in the blood, and that's not too surprising since this is delivered through the blood. In fact, miR34 levels are also increased in the combination, but not to quite the same levels as what we see in the full dose. We don't see any toxicity with this particular nanoparticle loaded with Let7, miR34, or the combination. What you can see here is that the levels of certain cytokines don't seem to be affected, and we've also done a panel of liver enzymes, and we see no problems with liver toxicity in this particular model. And in fact, the company MicroRNA Therapeutics that has now taken miR34 to clinical trials has done large animal studies which they've published on and see very low toxicity in, for example, monkeys that have been given this nanoparticle plus miR34. So this is a very busy slide, but I'll just point out the take-home message, and that is that the combination of miR34 plus Let7 is very effective against this particular aggressive model of lung cancer. So you can see here that the control animals have multiple nodules, and the nodules are large. The dual-treated animals have small nodules, and they have fewer of them. And that's all quantified over here. This is tumor burden, and this is tumor size. So while miR34 and Let7 alone each had some efficacy in this particular model, I should mention that this colored dot over here and square and triangle are these sections over here. So you can see I'm showing you really representative sections. This basically shows that Let7 and miR34 do show some efficacy when delivered systemically into this very aggressive, very resistant model of lung cancer, but the combination works most effectively. Our hope is that MicroRNA Therapeutics will start a phase 2 trial in lung cancer, and in fact they have agreed to open a trial at Yale in this coming year. And they will start it with miR34, but we will also do a phase 1 trial with the combination of miR34 plus Let7, just to see whether this shows any toxicity in humans. And the hope is that potentially this combination could be used in a phase 2 trial then for human non-small cell lung cancer. All right, in terms of survival, this experiment is still not quite finished yet, but what we find is that while Let7 doesn't seem to be by itself causing much of an effect in survival of this particular mouse model, miR34 and the dual, that is half the dose of Let7 plus half the dose of miR34, looks like it's extending the life of this particular aggressive model. And this blue arrow over here just indicates when we stopped the treatment. We stopped the treatment when half of the control animals were dead. And we don't yet know what might have happened had we continued treatment at this time. Potentially all of these animals could still be alive, at least in these groups. And we would love to do future studies where we take these experiments out more long term, not only to see whether we can see increased survival of these animals, but also whether we see some long-term toxicity that might emerge, and also some resistance mechanisms that might emerge in these treated animals. All right, so just to conclude this part, I've told you that we've developed methods for delivering tumor suppressor microRNAs like lead-7 and miR-34 into lung cells. And as you heard from Ronald earlier, we're only getting started in this field in terms of targeting these sorts of molecules to the right place and to the right tissues and to the right cell types within those tissues. And I imagine that in a few years, that smarticle is going to look really silly. It's going to be called a dumpsicle, and we're going to be talking about real smarticles, which will be able to be way more effective than what we have today. Just to reiterate, there's no obvious toxicity to this particular smarticle. Not only is the smarticle non-toxic, but we think that the microRNA mimics themselves are non-toxic. And one reason for that is that they're actually natural molecules. The body has had 1 billion years to learn how to get around off-target effects of lead-7 and miR-34. They're present in the last common ancestor of planaria and humans. So they've been around a long time. Normal cells have lots of them. They don't get affected by having more. But cancer cells and cancer stem cells, which have lost those particular microRNAs, do get affected when you give them more of the microRNA. And lastly, we've begun to explore whether combinations of microRNAs might be more effective. I think I've shown you at least one example where this is the case. But this is not the only case where you could think of using combinations. You heard from Ronald about potentially using combinations of drugs. We're thinking about using combinations of microRNAs and drugs. You can think of using other combinations of microRNAs. Another one that's very high on everybody's list you heard about from Ronald is miR-200. It's found deleted in many different cancers and would be a high priority for adding to this particular type of work. Okay, so for the second part, I'm going to talk about microRNAs as therapeutic targets. So this relies on the fact that in some cases, cancers become addicted to the overexpression of certain microRNAs. And so when the cancer is addicted or depends on that particular microRNA for its continued survival, if you can target the microRNA, you can cause the death of those cancer cells. So I'll give you the evidence for why we think that cancers become addicted to microRNAs first. Then I'll talk about different ways that we're coming at the problem of actually knocking out or knocking down the expression or the activity of these oncogenic microRNAs. And I'm going to end by talking about how we're trying to target these types of strategies or these types of molecules to the tumor microenvironment. This is something you heard about already from Ronit, and it's something that our lab is also very interested in doing. All right, so I'm going to introduce you to a microRNA called miR-155. miR-155 is an oncogenic microRNA. It's found to be upregulated in many different cancer types. I've already told you about how it's upregulated in non-small cell lung cancer, but it's most famous for being upregulated in almost every different kind of lymphoma and leukemia. And in fact, so this just shows germinal center lymphomas and different types of leukemias. It turns out that miR-155 is the active gene from the BcRNA, which my understanding is that Bic was discovered at the same time as Mic, but because there was no start codon or protein coding region in that RNA, it was ignored. We now know that Bic has a very potent oncogene, and that oncogene is in fact this microRNA miR-155. So Carlo Croce's lab showed that miR-155 was in fact an oncogene. If you expressed miR-155 in B cells by putting it downstream of the EU promoter, you can actually cause a B-cell-like leukemia and lymphoma to form in those mice. You can see, for example, that they have enlarged spleens, which is one of the sites of B and T cells in these animals. He likes to say that these animals look like they're obese, but in fact, no, they're not obese. They just have very large spleens, and that's what makes them look fat. So at the same time as Carlo Croce was doing these experiments, we were also trying to overexpress miR-155. In our particular case, we built a transgenic mouse that allowed us to integrate miR-155 in a single copy at the ROSA 26 locus, but in a way that allowed us to exquisitely control when and where it was expressed. So in our mouse model, at the ROSA 26 locus, we have inserted this particular transgene. It has the precursor of miR-155, so basically the stem loop of miR-155, downstream of a very strong promoter. This is the mini CMV promoter. But this promoter is repressible by doxycycline or tetracycline. So it has a TET operator site in the promoter. That allows us to switch this promoter off in the presence of tetracycline or doxycycline. Now between this promoter and the miR-155 gene is a lock-stop-locks cassette. So essentially, even if this promoter is being active, transcription stops in this particular region, and miR-155 is not expressed. In order to express miR-155, we need to add the Cre recombinase to the cells, which excises this stop cassette and allows now for expression through into miR-155, activating this particular gene. So you can see then how in the presence of Cre recombinase, we can turn on miR-155, and we can do this in a tissue-specific way because, as you know, there are tissue-specific Cre mice that you can cross your transgene to. And so once we've expressed miR-155 in cells, we can then turn off miR-155 by giving them doxycycline. And you can just feed it to them in their water or in their food, and it's good enough to get into cells and cause this particular promoter to now be inactive. So just to show you that the system works like we think it does, we can show that in the presence of Cre recombinase, we can actually see the recombination event that removes that lock-stop-locks cassette. We can also show that miR-155 levels increase in those animals. So if you cross them to a Cre mouse, in this case we've used the Nestin Cre mouse. The Nestin promoter drives Cre in the nervous system and in the hematopoietic system. We can see that miR-155 levels do get increased, for example, in the spleen. However, in the presence of doxycycline, we can show that, in fact, we can now switch off miR-155 levels again. So much like what the Croce lab showed, if we activate miR-155 in the hematopoietic system, we can see splenomegalia, this enlarged spleen. We also see enlarged lymph nodes, something that the Croce lab hadn't seen in their model. You can see this animal has almost a golf ball-sized lymph node growing out of the side of its neck. If you open up these animals, you can see that every one of the lymph nodes in this animal is in fact enlarged. Usually these lymph nodes are about the size of a pinhead. Here they're about the size of a gumball, so they've been enlarged almost 100 times. And in this particular mouse, it's a very penetrant disease. So at the time that we stopped counting these animals, about 2/3 of them had actually presented with some kind of lymphoma or some evidence of lymphoma, like, for example, paresis, which I'll show you later. These animals, the lymphocytes invade the entire animal, including the spinal cord, and that causes them to become paralyzed. We think this is a model for a human disease called diffuse large B-cell lymphoma. It's a very aggressive lymphoma, and in fact it's resistant to most kinds of therapy, meaning the patients with this disease ultimately die from the disease. There are some treatments, including R-CHOP, and I'll show you a little bit later, that we've tried a variety of that the mouse. And while this particular mouse model does respond quite nicely to CHOP, I'll show you that CHOP is in fact a very non-specific killer of all lymphocytes. So it leads to terrible side effects, both in the mice as well as in the patients. So in our particular model, you can see that while a normal spleen has this sort of white and dark pulp structure, we can see that in our mouse, these spleens just become completely effaced with lymphocytes. I can show you, I'll tell you later, these are B cells. And in fact, even in the thymus, which once again has light and dark pulps, these organs become completely effaced with B cells. Typically, the thymus just has T cells. I told you earlier that these lymphocytes can metastasize all over the body, and in fact here that you can see that they've gone into the brain and into the spinal cord. This is an example of a lymph node stained with Ki-67, which is of course a stain for proliferation, and you can see that this is a very aggressive tumor type. Every one of the cells, it's like 100% of the cells are staining with Ki-67, showing that they're all proliferating. This just shows you the liver of these mice, stained now with a B cell marker. This is B220, only expressed in B cells, and you can see B cells have metastasized into the liver as well in these animals. Needless to say, these animals all die, and I'll show you that in a few slides. Something that is interesting about this particular disease or this particular mouse model is that if you just look at the VDJ recombination that occurs in these B cells, in many cases we find that it looks like a single B cell is responsible for taking over the entire population of cells in that animal. So you can see monoclonal, for example, VDJ recombination events, suggesting that it's just one B cell somehow that has caused this particular tumor. And we don't understand how that could have happened yet. I should tell you that these cells are extremely aggressive. If you inject them subcutaneously into the flank of an immunocompromised mice, they grow these massive tumors within just a very short amount of time. I believe this is about a week of growth, and they get highly vasculated, as you can see here with this red look. Not only that, but the cells that are injected subcutaneously will metastasize all over this transgenic mouse, this host mouse. You can see that the lymph nodes in this host mouse also start to get enlarged. So you get lymph adenopathy, and we know that these cells came from the transplant because they contain the transgene. So this is a highly aggressive, highly metastatic disease. But we believe it is a good model for diffuse large B-cell lymphoma. So I think I've shown you that if you just turn on a single microRNA gene, in this case, miR-155, in our hands and in Carlo Croce's hands, we can show that those animals will develop cancer. So this proves without any measure of doubt that microRNAs can in fact be oncogenes. And the term has now emerged that these microRNAs should be called oncomeres as opposed to oncogenes. And I also told you that when we add doxycycline to these mice, we can see that miR-155 levels get switched off again. So we wondered if these tumors would depend on continued expression of miR-155 for their survival. And we were able to test that idea by allowing the animals to get tumors and then feeding them doxycycline and switching off the transgene, switching off or withdrawing miR-155 levels from those cells and asking what would happen to the tumors. And we saw another quite remarkable result. And that was that within just a few days of withdrawing miR-155 from these tumors completely regressed. Here's an example of an animal that within just one day you can see that these enlarged lymph nodes are starting to regress, and within 2 days the lymph nodes are almost back to normal size. Here's another case with a very large lymph node on the side of one animal, and within 4 days that lymph node has completely disappeared. While most animals that don't receive doxycycline will ultimately die. I've already mentioned that to you earlier. Every single animal that received doxycycline at this stage has survived. And in fact, these animals are still alive in some cases, and they haven't regressed back to having tumors again. So if you keep them on doxycycline, these animals will survive. So that told us that the tumors did in fact depend on miR-155 for their continued expression. Now that result was true both in the transgenic model and also in this transplant model. If you take cells from the spleen or from the lymph node of those transgenic animals and inject them subcutaneously into the mouse model, allow them to grow these tumors, and then feed the animals doxycycline, you can see that within just a few days, those tumors have regressed. So what this has given us, in fact, are two very nice models of diffuse large B-cell lymphoma where we can test the idea that targeting miR-155 could be a potential therapeutic for this particular disease. We can use either the transgenic mouse or this transplant mouse to test these ideas. Now, I should say that we do most of our work first in this transplant mouse for two reasons. First of all, it's very easy for us to quantify these tumors. We can actually see them growing on the side of the animal. We can measure them with calipers. And also, you can see that these effects are very fast. Just within a few days, we can see efficacy in this mouse. But in almost every case, we then follow up our experiments in the transgenic mouse. The transgenic mouse is a little more difficult for us to quantify the efficacy in because, for example, those enlarged lymph nodes are a little squishy, so you come in with calipers and you can just continue to well, essentially it's very difficult to accurately measure the volume of one of those lymph nodes. But I'm going to show you that in every case we can see some efficacy in both of the models. Okay, so you will, of course, realize that turning off a transgene with doxycycline like we just did, while it is an excellent way to give proof of principle that tumors become addicted to microRNAs and that the microRNAs could potentially be targeted, it's in no way representative of how human patients could be treated for this particular disease. First of all, human patients have a disease, diffuse large B-cell lymphoma, which is in fact poorly characterized, but does include overexpression of two oncogenes, MYC and Bcell2. And in fact, our mouse model also overexpresses both of those oncogenes. And in fact, that combination of MYC and Bcl2 overexpression makes diffuse large B-cell lymphoma classified as double hit diffuse large B-cell lymphoma, which is in fact the most aggressive and least treatable form of the disease. So our model is in fact a model for this even more aggressive form of diffuse large B-cell lymphoma. So we've asked, are there ways that we can target miR-155 as a potential therapy in this particular model? And as you've heard yesterday and earlier this morning, there are a number of different ways that you can target microRNAs in vivo using things like antagomers, which are essentially 2-O-methyl antisense RNAs that have a cholesterol molecule attached to them. You can use locked nucleic acids, and in fact, locked nucleic acids are the way that miR-122 is being targeted in hepatitis. I told you about a clinical trial that's already completed for targeting a microRNA that's involved in hepatitis replication. And the drug that was used to target miR-122 is in fact an LNA. We've actually shown that if you that you can deliver an antisense to miR-155 into the mouse model that I just told you about in the previous slide using a PLGA nanoparticle. It's like the PGA nanoparticle that you heard about this morning, except it has a lactic acid also thrown in there. And so we were actually able to deliver an antisense to miR-155 into that mouse model. However, none of these strategies works extremely well in our mouse model. We can see some efficacy, but the efficacy is not that great. And some of you will have had experience trying to transfect hematopoietic cells. B cells are notoriously hard to transfect, and they're notoriously hard to get drugs delivered to in vivo. And so we've basically asked around to see whether there are any other strategies that we might be able to use to target antisense molecules to the hematopoietic system. And as I said earlier, we've been working with a bioengineer called Mark Saltzman to help us design better nanodelivery mechanisms for the hematopoietic system. So the first thing that we've changed from the traditional methods is that instead of using nucleic acid type antisense molecules, which have a charged backbone, this phosphate backbone, like shown here for DNA and for RNA, we use a peptide nucleic acid backbone, which actually has a neutral charge. So it basically has a peptide bond here as opposed to a phosphate bond. We find that not only can we load more of these particular molecules into nanoparticles than we can other kinds of nucleic acids, if you will, because of this neutral charge, we can load almost 20 times more of these molecules into nanoparticles. But in addition, as I'll show you in a minute, this neutral charge makes it more easy for us to transport these molecules across membranes than does this charged backbone. That'll become important later on in the talk. Now, you've already heard about how tumors have leaky blood vessels. One thing that you might have heard about yesterday, sorry I wasn't here for that, is that in fact tumors have a particular microenvironment that makes them a little unusual. For example, they tend to use glycolysis a lot. They tend to make a lot of acids as a byproduct, and they have a slightly acidic environment. They're also hypoxic. They express certain markers on their surface, and you've already heard from Ronit about how you might be able to target nanoparticles and target drugs to tumors because of some of these features. She already mentioned, for example, using methods to maybe target the folate receptor, to target certain kinases, to target certain cell markers that are overexpressed in cancers. So we've instead focused on just targeting this acidity. So targeting the tumor microenvironment by looking for particles that are effective at low pH. And to do this, we teamed up with a biophysical chemist in the Department of Molecular Biophysics and Biochemistry at Yale, Don Engelman, who for years has been studying bacteria rhodopsin. Bacteria rhodopsin has a number of transmembrane domains, but one of its transmembrane domains has a very interesting feature, and that is that it's essentially able to cross membranes only at low pH. There are two charged amino acids within this transmembrane domain that need to be neutralized at low pH in order for this to actually pass through membranes. And he's studied this in intense detail. He knows that at normal pH, this peptide is unable to cross membranes. It can still bind to membranes, but it doesn't translocate across the membrane. At low pH, and by this I mean pH 6.5 and below, this particular transmembrane domain will now translocate across membranes, and in fact it carries its C-terminal portion into the cytoplasm of the cell. Now he has shown that you can attach a variety of different things to this portion of the peptide, or to this portion of the peptide, and he has used this particular peptide as a way to drag, for example, dyes that could target acidic parts of the organism. It can drag gold particles to tumors, for example. So one of my postdocs, Chris Cheng, asked whether if we attached this particular particle, this particular peptide, either to our nanoparticles or to our PNAs that would target miR-155, could we see tumor-specific targeting of this particular antisense molecule, which could give us a better specificity and potentially less toxic side effects than what we might have seen earlier. I should mention that FLIP, as it's called, PH-LIP, or pH lipid-inserting peptide, is not toxic as far as we know. In fact, it has been approved by the FDA for diagnostic purposes. It does not induce any membrane fusion or cause any holes in membranes. It doesn't cause membrane leakage. And in fact, when you raise the temperature again on these particular membranes, this peptide just pops right back out again and goes into the extracellular space. So what Chris did was he asked, what happens if we attach, okay, so I should tell you, I'm not going to talk to you today about attaching this particular peptide to nanoparticles. We have shown that it is effective at targeting nanoparticles to tumors. But we're still working on the best way to get those nanoparticles to release their contents into the cells. What I am going to tell you about today is that we can attach to the C-terminus of this peptide a PNA that targets an oncogenic microRNA. In this case, we're going to do miR-155, but we've also shown that this is true for other oncogenic microRNAs like miR-21, for example. And the idea here is that, sorry, let me just go back again. The idea here is that at low pH, this particular peptide will flip across the membrane and drag with it the antisense molecule to miR-155, which then becomes intracellular. Okay, so the first thing that Chris did was he asked whether this particular flip peptide would in fact target a dye to the tumors in our mouse models. Remember, we have these two models. One is the transgenic mouse that has these enlarged lymph nodes. And here you can see that flip will drag a 750 dye to that particular tumor in this mouse. It also targets the dye to the kidneys, which some of you will know is slightly acidic due to the uric acid and the like. And we are aware that any effects that we that we need to be cognizant of potential side effects in the kidney for any kind of therapeutic that arises from this technology. And in the second mouse, we can take these cells and we can transplant them into the immunocompromised mouse and cause these flank tumors. And in fact, we see the same thing, that flip directs the dye specifically to the tumors and not to other parts of the mouse. Very importantly for us, flip directs these anti-miRs, if you will, these antisense molecules, away from the liver. So it's one of the beauties of using antisense technology in liver disease that these particular molecules tend to accumulate in the liver. However, in our case, we want to avoid the liver. And what we can show is that if you add flip to those PNAs or LNAs or morpholinos, we've done it for many different things now, we can find that, in fact, the liver is not targeted. However, kidneys and the tumors are targeted. This is just another example of, in a second set of mice, just showing that within just a few hours, you can see that the dye gets targeted to the kidneys, but tends to accumulate only in the tumors. So some of it will get cleared through the kidneys, but it does accumulate in the tumors. This just shows you that the drug does get cleared. It does get cleared through the bladder. So there might be some issues here with long-term retention, but we do see that within 72 hours and within 96 hours, we still see some targeting in the tumors. All right, so in this case, what Chris has done is he's designed a PNA, a peptide nucleic acid, that has the antisense sequence to miR-155, and he's added it to flip through this disulfide linkage over here, such that at low pH, flip will drag across the membrane this anti-miR-155 PNA, and that it gets reduced in the reducing environment of the cytoplasm to release the PNA, which can then go and find its target miR-155. So before we tried any experiments in vivo, Chris was able to show that in fact this does happen, that miR-155 does in fact get internalized and does in fact knock out the expression or the activity of miR-155. This just shows you that we can actually create this dimer, the PNA flip dimer. Okay, so flip PNA, when tagged with a TAMRA tag, in fact, does enter into cells. This is this red dye over here. It enters into cells only at low pH, but not at normal pH. When it gets into cells, it in fact seems to function to knock out miR-155, because if we look at expression of a luciferase reporter gene that has a perfect miR-155 complementary site in its 3' UTR, only when we deliver the flip PNA at low pH do we see an increase in reporter gene expression, indicating that we've knocked out the expression of the microRNA that's regulating this reporter. Okay, so what happens in animals? I told you that we have these two models. I'm going to start by telling you what happens in this model, where we can take miR-155 overexpressing lymphocytes and inject them subcutaneously, and then I'll tell you what happens in the transgenic mice that have these enlarged lymph nodes. All right, so in the first case, what we've done is taken cells from the transgenic mouse and established flank tumors in these immunocompromised mice, and then treated them over the course of a few days with a number of different reagents. In the first case, we've used our experimental reagent. This is the flip anti-miR-155. And we've compared that to just a flip anti-scrambled sequence, a PNA that just has a scrambled sequence. We've also compared that to the PNA to miR-155 alone, without the flip attached. And then as a positive control, we've used doxycycline, which I've already shown you causes the death of these cells. Compared that to a mock-treated animal. These animals are just treated with saline. And then, they're treated with the vehicle for CHOP. Now, CHOP is the best current therapy for human diffuse large B-cell lymphoma. I'm not going to try and tell you what CHOP is, but it's a combination of 4 different drugs. And then we wait a few days and we analyze these animals for tissue distribution of the antisense, as well as for efficacy against the cells. Okay, so one thing that you can see immediately is that these flank tumors grow much more slowly if we treat them with the flip PNA, as opposed to the tumors treated with any of the controls. Now, one thing you'll notice here is that these tumors have not completely regressed. So if you treat these animals with doxycycline, for example, we can see that the tumors start to regress. So we're still not seeing 100% efficacy in this particular model. But one thing I should point out is that we're using very low doses of flip PNA here. Typically with an antisense type experiment that you might have heard about before, and certainly for the clinical trials for the anti-miR-122 in hepatitis, most of these antisense molecules are delivered at about 100 milligrams per kilogram of the animal. We're using, in this case, only 1 milligram per kilogram, so 2 orders of magnitude lower than what you would typically use with an antisense type experiment. And we're still seeing some efficacy. We hope that once we can ramp up production of this particular molecule, we can get to higher doses, we should see even greater efficacy. One thing that is shown here is that CHOP is also effective at causing tumor regression in this particular model. I'm going to show you in the next slide that unfortunately CHOP does this very non-specifically. It just kills all lymphocytes in this particular animal. Okay. So even though we don't see complete tumor regression in this model, one thing that we do see is a complete loss of metastasis in this model. So if we inject those cells subcutaneously, I told you that they will over a short time, within a few weeks, metastasized to all different parts of the body, including the spleen. What we find is that in animals treated with the anti-MIR-155 flip, there is very little evidence for metastasis in this model, unlike in the control-treated animals. Sorry, but this mouse is not very responsive. Okay, there you go. In the control-treated animals, let's see extensive metastasis. This is the evidence that just shows you that if you just do a total blood cell count in these animals and just count the total number of lymphocytes, you can see that CHOP causes about 10-fold excess lymphocyte cell death, so just non-specifically killing lymphocytes. And that's why it's effective as a therapy, but also why you get such horrible side effects with this particular drug. So our flip MIR-155 had the same general low toxicity that you see when treating these animals with doxycycline. And the scrambled and the anti-MIR-155 animals had the excess lymphocytes that we see that I've reported earlier. This is just showing you that if you section through the livers, you can see very little evidence of B-cell metastasis to the liver. This is now stained with that B220 antibody. In addition, the animals treated with the flip PNA live longer than animals treated with what up to this point is one of the gold standards for antisense technology, and that's this LNA technology, or the locked nucleic acid technology. So we see an increased survival in the animals that have been treated with the flip anti-MIR-155, certainly compared to the mock treated, but also to the animals treated with an LNA. So what about in the transgenic mouse now, which is an immunocompetent mouse? It takes a little longer to present with disease, up to 2 or 3 months, but ultimately every animal will present with disease and will ultimately die of the disease. So in this case, I mentioned to you it's a little harder for us to measure efficacy. First of all, the tumors are forming inside the mouse, and we cannot readily detect them just by looking at the mice. However, at some point we do detect these enlarged lymph nodes. We also notice that these animals become scruffy, and they lose the use of their hind limbs. So they drag their hind limbs around. Ultimately, they become paralyzed, cannot feed, and will die. Okay, so in this case, what Chris has been able to do is wait until the animals present with some evidence for lymphoma, and then administer by IV into the tail vein either flip anti-MIR-155 or the anti-MIR-155 PNA by itself, or just a mock treatment. And he waits 2 to 5 days and then analyzes these animals. So first of all, he was able to show that in fact the anti-PNA by itself, when attached to a TAMRA tag dye, does not really target tumors very, very well. However, the flip PNA version targets tumors very well. Secondly, he was able to show that the anti-MIR-155 PNA accumulates in the liver, much as what other people have shown with PNAs. However, the flip tagged anti-MIR-155 avoids the liver. Okay, so this is one example. He's done this now in a number of different mice. You can see that this mouse presented with this enlarged lymph node, and within just 3 days you can see that a single injection of the flip PNA into this mouse caused that tumor to regress. Now, even though this is a dramatic visual, it's kind of hard for us to quantify, because like I said, calipers here, in fact, we're unable to get a volume of the tumors using calipers, but just by eye you can see that these tumors have regressed. Now, I was hoping that these movies would play. I'm not too sure whether they are going to play. In fact, I'm pretty certain that this movie will not play. What I'm going to do is, I've got some other movies which I know will play, but I'm going to just tell you that this is an animal that, if this movie was to play, is essentially on death's door. The vet had told us to come in and euthanize this mouse for humane reasons. It was essentially not moving. You can see it's dragging its hind limbs. This is Chris's finger here. He prods the mouse does not really move. He thought, is it even worth wasting some of my precious molecule by injecting this particular animal when it looks like it's going to die? As it turns out, this movie, if it was to play, shows this animal 5 days later after 2 injections of the flip antimirror, and it's running around the cage. So I will hopefully be able to show you those movies later on. They are on this computer, but they're just not playing right now. This is another example of a mouse. Sorry about the noise. You can see this animal is dragging its hind limbs. It's not very sprightly. Within just a few days of getting an anti-MIR155, this animal is now walking pretty much normally. So Chris has analyzed these mice in more detail. He's done histology on these mice. He's done immunotyping on these mice. We can show that the Bcell count in these mice goes down. They start to get more T cells when they had mostly B cells. He's also shown that there's very limited toxicity that's observable in these mice. Now we think that these lymph nodes are regressing by apoptosis because we can see increased tunnel staining in the animals treated with the flip antimirror compared to the animals treated with just the anti-PNA by itself. We also find that some of the genes that we know get altered in the expression in this particular tumor model also change the expression when the animals are treated with the flip anti-MIR155. So one way that we managed to identify the genes that were turned on and off in this tumor model was by doing gene expression analysis, where we compared tumors as they were growing to tumors that were regressing in the presence of doxycycline, and then looked at differential expression. So which genes got turned on, which genes were turned on during tumor progression that were then turned off again when the tumors regressed. I should tell you that on this list are our 2 favorite oncogenes, MYC and Bcl2. They both get dramatically upregulated in these tumors. Dramatically upregulated in these tumors, but downregulated as the tumors are regressing. In contrast, a number of tumor suppressor genes get downregulated as the tumors are progressing and then upregulated again when the tumors are regressing. If you just look at target enrichment of the genes that change in this model, you can see that the greatest number of genes that change have been associated with cancer. This just shows you some of the tumor suppressor genes that change their expression or they get upregulated upon MIR155 withdrawal. And on this list are a few genes that you'll recognize, things like p53 interacting protein, BRCA1 interacting protein, MAFB. BAC1 has been shown by a number of labs to be a direct target of MIR155. In fact, all of these genes, we believe, are in fact direct targets of MIR155. So of the 740 genes that are predicted to be MIR155 targets, 51 also changed their expression in our particular mouse model. Okay. And in fact, if you just validate that with qRT-PCR, you can see, for example, the BAC1 levels will rise when the animals are treated with doxycycline. This basically shows qPCR for not only BAC1 but for other members of that list like SMARCA4, HIVEP2, MAFB, etc., and p53 interacting proteins. So they all look like we can validate these as direct targets of MIR155 that get re-expressed when MIR155 levels go down. Now, when we treat the animals with the anti-MIR155, I showed you that these tumors will regress. If you harvest these animals here, we can still get them at a time when they're still when the lymph nodes are not completely have not completely disappeared. We can find that some of these tumor suppressor genes get re-expressed in those animals. Here's BAC1 and MAFB. So we think that when MIR155 levels are reduced in this particular treatment regimen, these tumor suppressor genes get re-expressed, and that leads to regression of the tumors. So we also showed that there was very limited toxicity in these mice. Okay, I'm almost done. Yeah. So this just shows you sections through the kidney, for example. We can see a normal kidney profile, the normal level of certain liver and kidney enzymes within the normal range. Okay, so just to summarize this portion of the talk, I've told you that tumors can become addicted to microRNAs. We've shown this now for a number of microRNAs, not just miR-155. So this is important because it makes these microRNAs very good targets for anti-cancer therapy through the sorts of methods that I've told you about and that you've heard about at this workshop. So we delivered an anti-miR delivery system that we think avoids common barriers to anti-miR, both tissue-specific barriers as well as cell-specific barriers. So we can deliver PNA anti-miRs to cultured lymphocytes, which is a very difficult thing to do, and to solid lymphoid tumors in vivo. We can target these antisense molecules to inhibit oncogenic microRNAs, and we can show therapeutic efficacy in vivo. And we've shown that it looks like they work by reactivating certain tumor suppressor genes that had been targets of these oncogenic microRNAs. So I'll just end by saying that nanoparticle delivery mechanisms are being developed now that are overcoming some of the traditional barriers that have scared away a lot of biotech companies from moving in this particular area. We think that not only are they effective, but they don't have very much toxicity. And so I think I just mentioned this, but delivery issues are being overcome with next generation nanotechnologies, some of which you're going to hear about at this particular conference. And it's my hope that some of these molecules will ultimately make it into human clinical trials and ultimately be medicines for some of these diseases. So just to thank some of the people in my lab who did the work. The lung cancer work was mainly done by Andrea Kaczynski, who now has her own lab at Purdue University in Indiana. Chris Cheng is a postdoc in my lab who did the FLIP anti-miR-155 work. Anybody here shown in red had some hand in some of the experiments that I told you about? Either they developed the mouse models or they started some of the work. I have to thank our collaborators, without which none of this would have been possible. Some of these groups for funding, and I'm happy to take any questions if there's still time. Thanks.